Hi, my name is Bernd Hänfling and I would like to present the latest output from our work on UK lakefish monitoring using eDNA metabarcoding. This work was initiated by the UK Environment Agencies and involved many different colleagues. Only the key authors of the results presented here are mentioned on this slide. The rationale for developing an eDNA-based method was quite simple. Because of the many practical and legal constraints around using standard gill netting approaches in the UK, we don't have a fully operational WFD method based on lakefish, which is on the other hand a legal requirement under European Water Framework Directive. The method details from what I'm presenting now are published in the papers listed here, but important here to mention is that our approach is based on collecting lots of different samples from each individual lake. This allows us to use so-called site occupancy as a semi-quantitative measure of fish abundance, so we don't need to rely on the number of sequences we find for each species, but we can use a number of individual sites within the lake where each species was detected. The method we have developed was optimized to characterize fish communities on a lake basis, so to provide lake-wide biodiversity estimates rather than site-specific ones. It is based on collecting 22-liter samples from the shoreline during the winter months, which has shown to be the most effective and efficient strategy based on the initial development work of some of the UK's larger lakes. For example, you can see from the sample rarefaction curves from Windermere on the bottom right that in winter fewer samples are required compared to summer, and that eDNA is actually more homogeneously distributed in winter. The graph on the left shows combined site occupancy data from a number of large lakes. So you can see for almost all of the 34 species we looked at there, winter detection probability was higher from the shore compared to the offshore samples, or approximately equal. We have used this approach to collect data from 101 water bodies, which were representative for UK lake types, size and geographical spread. This included lakes of different alkalinity and depths and lake sizes ranging from 4 to over 700 hectares, including the UK's largest lake. In total, across the entire data set, we found 40 fish species, of which 16 were rare and occurred only in less than 5% of all lakes. The fish diversity in the UK is relatively low compared to continental Europe, and although the highest diversity, species diversity was 25 species, the median was only 7 species per lake. So our pilot studies showed that the sample size we used should be sufficient to capture most species, but we also validated this against the full data set. So we carried out a sample coverage analysis, which is a rarefaction and extrapolation method to predict the completeness of species richness sampling at different sample numbers. And importantly, it is scaled to 100%. So that means the results are directly comparable among lakes of different total richness. So for analysis purposes, we have chosen an arbitrary user-defined threshold of 95% sample coverage. So this is the point where 95% of species richness of a lake is identified. Our analysis showed that 20 samples, 96% of our lakes achieved this threshold, but this dropped actually only slightly to just under 90% when we reduced the number of samples to 10 per lake. This means that a fairly good representation of species diversity can be achieved with much fewer samples than we have used, and this might be sufficient for many purposes. We also carried out some simple rank correlations to explore which different factors might influence the number of samples needed to achieve the 95% threshold. So the most obvious one to test was lake size, but this turned out not to be correlated with sampling effort. However, we did find a positive correlation with total species richness and a strong negative correlation with mean occupancy, which is a measure of, I guess, average fish density. So the highest effort is required in lakes which show a high species diversity but low fish density. One very important finding from the development phase of our project was that eDNA abundance in lakes is quite closely related to fish abundance. You can see this in these graphs here, where we plotted eDNA site occupancy against species rank abundance based on long-term data in two connected lake basins, Windermere North and Windermere South Basin. We were able to assign each species an abundance ranking from 1 for the most abundant species to 16 to the least abundant species. In both cases, the correlations were highly significant and strong and consistent across 3 different sampling campaigns over 2 years. We found similar relationships really in all other lakes we looked, and site occupancy seems to be generally a very good measure of relative abundance for lakes, at least of the size range we've included in this study. So the data that we can extract from eDNA metabarcoding give us a really good semi-quantitative representation of the fish community structure. The analysis I'm showing here demonstrate that the data are also ecologically meaningful. So here we looked at 14 lakes in Wales and Northwest England, which we had pre-classified into 3 types based on alkalinity, depth and distance to sea. So we were expecting different fish communities in these different categories. When we carried out a cluster analysis based on eDNA fish community dissimilarity, you can see that the types largely cluster together, apart from type 3, which was split into 2 clusters. But this could be explained by the fact that the two lakes which form a separate cluster were highly manipulated by stocking, so additional species were introduced there. So in order to evaluate how sensitive these data are to sample size, or these semi-quantitative data, we've carried out a number of resampling analyses, but I'm really only showing one of them here. Instead of full resampling, such as bootstrapping, here in this case we split the data set of 20 samples in half into 2 smaller data sets of 10, while still maintaining the full spatial coverage of the lake. So it's not a random resampling. So it was important that it is still the same as representative as the full data set. We then carried out a multidimensional scaling analysis based on Bray-Curtis dissimilarity of all full and half data sets, which you can see here. So in this figure, all the full and half data sets from one lake are circled. And you can see clearly that in most cases, the subsets are much closer related to each other and to the full data set from the same lake than to a different lake. So this gives us some confidence that semi-quantitative community structure signal is quite robust to reducing the sample number from 20 to 10. Of course, one thing eDNA cannot provide us with is absolute abundance, at least not at the moment, or age class information for different species, which is often used in methods which use fish data to assess the quality of water bodies. The question was now, can we detect anthropogenic pressure across a wide range of different lakes, just relying on these relative abundance data which we can extract from eDNA, especially in a situation where like the UK, where we actually have got very species poor lakes. So are the data powerful enough that you can actually assign a lake to WFD status? So we've used our data sets of this 101 lakes to explore this question. For all lakes, we had pre-existing WFD classifications based on a number of different assessments methods, so not fish. So you can see this on the left-hand side here. We then explored whether various metrics which we could extract from the semi-quantitative eDNA metabarcoding dataset would show response to quality status. And this analysis was carried out by Nigel Willby and colleagues at the University of Stirling. Importantly, this analysis was done using a surrogate of natural productivity. The so-called morphoedaphic index is a covariate of our anthropogenic pressure. So the most informative measures we could identify from our data sets were the site occupancy of trout, perch, roach, all other salmonids grouped together, and the combined values for carp and common bream. So based on these data, we could then calculate the predicted values for these metrics given a particular morphoedaphic index, and consequently an ecological quality ratio. We then defined class boundaries for four of the five WFD categories to optimize the agreement with the pre-existing WFD classification of these lakes. And you can see the results of this in the graph on the left-hand side and the quantification of this agreement in the table on the right. So the overall we achieved a pretty good agreement of 99% of all lakes were in agreement plus minus one class, and 54% were exactly the same class. The figure here shows this comparison on a map for the classification of lakes based on the eutrophication sensitive BQEs on the left hand side, and that based on fish eDNA on the right. We also had a look at the sensitivity of this metric to sampling effort. So for this, we used again our 10 samples subsets for this. So you can see that in most lakes, the EQR is only very mildly affected by sample number reduction. Only in a few cases, the reduced 10 sample datasets resulted in a shift in WFD classification. And those are largely lakes which are right on the border between the two categories anyway. So I want to finish with showing the many people who have contributed to this research, and I'll take any questions if there's still time.